human genetics. So human genetics is, we have learned a part of this in the mutation section itself. We have learned a few things about the chromosome, what is happening to chromosome, etc. So keep those things in mind and recall all those we learned in that session. So human genetics means genetics was actually it was by the when we hear the name genetics, the first person's name who comes to our mind is Gregor Mendel, a person who conducted so much experiments in pea plants. And we know dwarfism, the recessive character, the tall plant, and this Mendel's name is the first name which is coming to most people's mind when we start speaking about genetics. And there are few laws which was proposed by genetics like law of dominance, law of independent assortment, law of segregation, etc. Now, in human beings, basically, there are 23 pairs of chromosomes. We have 23 pairs of chromosomes, out which 22 are autosomes and one pair of sex chromosomes. And this is the basic chromosome structure. One pair of sex chromosomes. And this here you can see 22 pairs. 1, 2, 3, 4, 5, 6, 7, 8 and 23 pairs of chromosomes you can see here till 22, then one pair of sex chromosome. This is what I said. Now, a basic understanding of where this chromosome is located. You all are aware that in a cell, if this is the cell, you will be having a nucleus, and inside the nucleus the chromosomes are located, and the chromosomes and inside the chromosomes the chromosomes are located, and inside the chromosomes the DNA is tightly packed. This is how you can see here chromosome. A chromosome is located here, and inside the chromosome the genes are located, and specific parts of the chromosome in a of DNA sequence in a chromosome is called a gene. Now, next is the what a karyotype and a ideogram is. We have, I said, we have 23 pairs of chromosome, and we have, if we have 23 pairs of chromosome, it is arranged. When we show a picture, this is how it is being. The first picture itself, you understood it is arranged in a particular manner. Take. This is what is known as a chromosome. That is, the chromosome is arranged. The term karyotype is the total number of chromosome constitution of a organism is arranged in a particular order, and that is what is known as karyotype. So it is the group characteristic of an chromosome is taken into consideration. The particular chromosome of an individual is the chromosome size, and the arrangement of the chromosome according to its size is known as karyotype. That is what is known as karyotype. That is, the total chromosome constitution of an organism is arranged in a particular manner. For doing karyotype means ഒരു ഓർഗാനിസത്തിന് അല്ലെങ്കിൽ ഹ്യൂമൻ ബീങ്ങിന്റെ ഏത് ഓർഗാനിസം, പ്ലാന്റ് ആണെങ്കിലും അനിമൽ ആണെങ്കിലും അതിന്റെ ബോഡിയിലെ ക്രോമസോംസ് മൈക്രോസ്കോപ്പിലൂടെ നോക്കിയതിന് ശേഷം അതിനെ ഒരു അറേഞ്ച്മെന്റ്, സെല്ലിനകത്ത് ഇറ്റ് ഇസ് ആക്ച്വലി നോട്ട് ലൈക്ക് ദിസ്. Karyotype means നമ്മൾ for interpretation and for analysis and all we arrange in a particular manner like this. Karyotype എന്ന് പറഞ്ഞാൽ അതൊരു പിക്ടോറിയൽ റെപ്രസെന്റേഷൻ മാത്രമാണ്, അല്ലാതെ and that is karyotyping എടുക്കുന്ന കാര്യങ്ങൾ. 1. Size of chromosome is taken into consideration. Position of centromere. Centromere is, I'll come to it later, but still centromere is the middle portion of the chromosome is called centromere. So, the size of the chromosome is taken into consideration. Position of chromosome is taken into consideration. Then the presence of secondary construction, that is what is here. The centromere or the construction is taken into consideration, and size of the satellite chromosomes in the chromosome is taken into consideration. So, karyotyping is the total chromosome constitution. This is an picture. The picture which is shown here, this is a karyotype. ഇതിനെയാണ് ഒരു karyotype എന്ന് പറയുന്നത്. So karyotype is the total chromosome constitution of an individual or chromosome makeup of a species. That is known as karyotyping. Now, next is the term karyotyping is derived from the word karyon, which means nucleus. Nucleusനകത്തുള്ള constituentsന്റെ details പറയുന്നത് കൊണ്ട് it is karyo means karyotype. We know eukaryote and prokaryote. So karyo means nucleus. Then karyotype is represented as ideogram. ഈ karyotypeനെ അറേഞ്ച് ചെയ്തതിനു ശേഷം അതിനെ പിക്ടോറിയൽ ആയിട്ട് റെപ്രസെന്റേറ്റ് ചെയ്യുന്ന അല്ലെങ്കിൽ ഡയഗ്രമാറ്റിക് representation of this karyotype. The diagrammatic representation of karyotype is called ideogram. That is the main difference. Karyotype is the total chromosome complement of an individual. The diagrammatic representation. See here, karyotype ഇലാണെങ്കിൽ എന്താണ്, chromosome ഇങ്ങനെ ചെരിഞ്ഞിരിക്കും, ഇങ്ങനെ വളഞ്ഞിരിക്കും. It is like this when we draw a karyotype. But when we draw a ideogram, it is a perfect pictorial representation. See the difference between this is a ideogram and the previous one which we saw was this is a karyotype and this is a ideogram. So when the haploid set of chromosomes of an organism is arranged in the order of decreasing size, that is known as ideogram. So karyotype, total chromosome constitution. Then ideogram is the pictorial representation of the chromosome. Now let's see the chromosome complement. So how the karyotyping is done? Karyotyping is, there are some rules which is followed while making a karyotype. While making a karyotype, you have to arrange the chromosome in the decreasing order of size and centromere in a straight line. The main thing is decreasing order of size. The chromosome is arranged in the decreasing order of size. So in that picture we have noticed extreme, if this is a karyotype, the longest chromosome will be at the extreme left. It's like this based on the length, and the shortest chromosome will be at the extreme right. The shortest chromosome will be at the extreme right, and the longest will be on the extreme left. The longest chromosome on extreme left and shortest chromosome on extreme right. Then sex chromosomes or allosomes. Allosomes are also called sex chromosomes, are arranged on the right side. Here itself the sex chromosome or allosomes will be arranged. Now, ഇപ്പോൾ കാരിയോടൈപ്പിൽ രണ്ട് കാര്യങ്ങൾ. ഒന്ന്, മാക്സിമം ലെങ്ത് ഉള്ളത് ലെഫ്റ്റിൽ, എക്സ്ട്രീം ലെഫ്റ്റിലും, ഏറ്റവും ചെറുത് എക്സ്ട്രീം റൈറ്റിലും. ഇനി എക്സ്ട്രീം റൈറ്റിലെയും ഏറ്റവും അറ്റത്ത് വരുന്നത് അല്ലോസോംസ് അല്ലെങ്കിൽ സെക്സ് ക്രോമസോം. അതായത് ഇവിടെ വരുന്നത് വിൽ ബി അല്ലോസോംസ്. അല്ലോസോംസ് ആണ് എക്സ്ട്രീം റൈറ്റിൽ വരുന്നത്. ഷോർട്ടസ്റ്റ് വിൽ ബി ഹിയർ, ദാറ്റ് ഈസ് ഓൺ ദ റൈറ്റ്. Now, advantages of karyotyping. What is the purpose of actually doing this karyotyping? എന്തിനാണ് ഇങ്ങനെ ചെയ്യുന്നത്? It reveals the structural features of each chromosome. Each chromosomes, the structure can be studied if you arrange the chromosome based on its length. It helps in the structural arrangement of chromosomes. Then, it helps in studying chromosomes banding pattern. Now, banding pattern is based on the staining technique. Banding pattern is based on the staining technique. So, the advantages of karyotyping are: it reveals the structural features of each chromosome. It helps in studying chromosomes banding pattern. Then it helps in the identification of chromosomal aberrations. Chromosomeന്റെ structural എന്തെങ്കിലും വ്യത്യാസം വരുന്ന എങ്കിൽ, chromosomal aberration we have learned in mutation, structural wise, if there is any change in the chromosome, that's known as structural aberrations. Numerical aberrations, etc. are there. Then diagnosis of prenatal defects. This is the most very important. So, the diagnosis of prenatal defect, genetic defects, is done with the help of this karyotyping. Then, and they are used in studying the evolutionary changes. So, these are the advantages of karyotyping. 1. It reveals the structural features of chromosome. 2. It helps in studying the chromosome banding pattern. Actually, banding pattern we'll discuss in a short while. It is the, after staining, the chromosome will have some patterns on it. That's known as banding pattern. And it helps in the identification of structural aberrations. That is, if any mutation has happened. Diagnosis of prenatal genetic defects. Then it helps in studying the evolutionary changes. Now, next is chromosomal banding. Chromosomal banding is to study. This is what is known as band. You can see here one band, another band, another band here. There are many bands. So, these bands appear on the chromosome when it is subjected to staining. There are different types. Chromosome banding means the chromosomes are identified, which chromosome, which part of the chromosome, or we can identify which gene, that is based on the response of a chromosome to stain. That is what is known as chromosome banding. Chromosome stain cheyana. We have, and some of the stain which we have, you might have used will be in the botany laboratory or in zoology. You have acetocarmine, you use methylene blue, അതുപോലെ ഓരോ സ്റ്റെയിൻസും സ്പെസിഫിക് റീജിയൻസിൽ ബൈൻഡ് ചെയ്യാൻ വേണ്ടിയിട്ട്. അങ്ങനെ ക്രോമസോമിന് സ്പെസിഫിക് ആയിട്ടും അങ്ങനെ ദർ ആർ സം സ്റ്റെയിൻസ്. ബാൻഡിങ് ടെക്നിക്സ്. ഇപ്പോൾ ഈ ക്രോമസോമിനെ പലതരത്തിൽ സ്റ്റെയിൻ ചെയ്യാറുണ്ട്. വൺ ഇസ് with the help of this quinacridine. This is a stain. Giemsa is another stain. Then these are the two banding pattern which is given in your textbook. നിങ്ങളുടെ ബുക്കിനകത്ത് കൊടുത്തേക്കുന്ന രണ്ട് പാറ്റേൺ ഇതാണ്. ക്വിനാക്രിൻ വെച്ച് സ്റ്റെയിൻ ചെയ്യുമ്പോഴാണ് you will be getting the Q banding എന്ന ടെക്സ്റ്റിൽ കൊടുത്തേക്കുന്നത്. Q banding. Q banding means ക്വിനാക്രിൻ വെച്ച് സ്റ്റെയിൻ ചെയ്യുമ്പോൾ ക്രോമസോമിന്റെ ചില ഭാഗത്ത് ബൈൻഡ് ചെയ്യും. അതിനെ Q banding എന്നും G banding എന്ന് വെച്ചാൽ GMSA സ്റ്റെയിൻ വെച്ച് G banding means GMSA stain. ഇത് രണ്ടും ക്രോമസോമൽ സ്പെസിഫിക് സ്റ്റെയിൻസ് ആണ്. അപ്പോൾ ആ സ്റ്റെയിൻസ് വെച്ച് സ്റ്റെയിൻ ചെയ്യുമ്പോഴേക്കും ക്രോമസോമിന്റെ ചില ഭാഗങ്ങൾ സ്റ്റെയിൻ ചെയ്യപ്പെടും. ആൻഡ് ദാറ്റ് ഇസ് വാട്ട് ഇസ് നോൺ ആസ് Q banding ആൻഡ് G banding. ഇനി വേറെ തരം സ്റ്റെയിനിങ് ആണ് NOR, അതായത് ജീനിന്റെ ക്രോമസോമിന്റെ NOR സ്പെസിഫിക് ലൊക്കേഷൻസിൽ ബൈൻഡ് ചെയ്യുന്നത്, സെൻട്രോമെറിക്. Now, banding technique. ഒരു ടെക്നിക്ക് ജസ്റ്റ് ഞാൻ പറയാം. ക്രോമസോം ഇപ്പോൾ നമ്മൾ RBC ഐസൊലേറ്റ് ചെയ്യുന്നു. Then you isolate the cells. Then chromosome is stained with the quinacridine mustard stain. It will be this is actually a stain technique. Basically how it is being done. This slide just shows. You need not study the details. But just I am showing how it is being done. Chromosome എടുത്തതിന് ശേഷം സെൽസിൽ നിന്നും ഐസൊലേറ്റഡ് ആയിട്ടുള്ള chromosomes or under the on the slides it will be stained with the quinacridine mustard stain. അതാണ് Q banding. Quinacridine mustard stain is responsible for Q banding pattern കിട്ടുന്നത്. Q banding. And it is subjected to UV light, and the banding pattern will be based on region rich in AT bases and region rich in GC bases. This is the chromosome. ഇതാണ് ക്രോമസോം എങ്കിൽ ഒരു സ്ഥലം ഡാർക്ക് ആയിട്ട് സ്റ്റെയിൻ ചെയ്യും, ഒരു സ്ഥലം ലൈറ്റ് ആയിരിക്കും, അടുത്ത സ്ഥലം വീണ്ടും ഡാർക്ക് ആയിട്ട് സ്റ്റെയിൻ ചെയ്യും, അടുത്ത് വീണ്ടും ഡാർക്ക് ആയിട്ട് സ്റ്റെയിൻ ചെയ്യും. Another region will be darkly stained, and this another region again it will be darkly stained, and these are called light bands. These two are dark bands. മൈക്രോസ്കോപ്പിൽ കൂടെ നോക്കുമ്പോൾ ചില ഭാഗങ്ങൾ ഡാർക്ക് കളറിലിരിക്കും, ചില ഭാഗങ്ങൾ ലൈറ്റ് കളറിലിരിക്കും. അതിനെയാണ് ഡാർക്ക് ബാൻഡ്സ് എന്നും ലൈറ്റ് ബാൻഡ്സ് എന്നും പറയുന്നത്. These are the dark bands and these are the light bands. Light bands. Keep always in mind mutations I have taken. Light bands. അപ്പോൾ region rich in 80 bases. ഈ ഡാർക്ക് ബാൻഡ്സിൽ അഡെനിൻ തൈമിൻ റെസിഡ്യൂസ് AT റിച്ച് റീജിയൻ ആയിരിക്കും. AT അല്ലെങ്കിൽ അഡെനിൻ തൈമിൻ റെസിഡ്യൂസ് കൂടുതലായിരിക്കും ഇൻ ദ ഡാർക്ക് റീജിയൻ. ബട്ട് ഇൻ ദ ലൈറ്റ് റീജിയൻ GC rich, guanine cytosine will be rich, or GC will be rich in light stained region and AT will be rich in dark stained region. This is a basic what is എന്താണ് banding എന്ന് മനസ്സിലാകാൻ വേണ്ടിയിട്ട് ഞാൻ പറഞ്ഞതേ ഉള്ളൂ. Then it is exposed to fluorescence for observing under the microscope. അപ്പോൾ ക്രോമസോമിൽ ക്വിനാക്രിൻ മസ്റ്റാർഡ് സ്റ്റെയിൻ ചെയ്യുന്നു, അതിനെ UV ലൈറ്റ് എക്സ്പോസ് ചെയ്യുന്നു, അത് ആ സ്റ്റെയിൻ ഫ്ലൂറസെൻസ് ഉള്ളതുകൊണ്ടാണ്. ദെൻ രണ്ട് ഭാഗമായിട്ട് നിങ്ങളിപ്പോൾ എത്ര ഓർത്തിരുന്നാൽ മതി, ക്രോമസോമിന്റെ ചില ഭാഗങ്ങൾ ഡാർക്ക്ലി സ്റ്റെയിൻഡ് ആവും, ചില ഭാഗങ്ങൾ ലൈറ്റ്ലി സ്റ്റെയിൻഡ് ആവും. ഡാർക്ക്ലി സ്റ്റെയിൻഡ് ആകുന്നതിനെ ഡാർക്ക് ബാൻഡ്സ് എന്നും ലൈറ്റ്ലി സ്റ്റെയിൻഡ് ആകുന്നതിനെ ലൈറ്റ് ബാൻഡ്സ് എന്നും പറയും. ഇനി ഡാർക്ക് ബാൻഡ്സ് എന്ന് പറയുന്നത് എന്താണ്? AT റിച്ച് റീജിയനാണ് ഡാർക്ക് ബാൻഡ്സ്. യു ക്യാൻ ജസ്റ്റ് റൈറ്റ് ഇറ്റ് ഡൗൺ, ഇറ്റ്സ് നോട്ട് ബീങ് ഗിവൺ ഇൻ യുവർ ടെക്സ്റ്റ് ബുക്ക്. AT ബാൻഡ് എന്ന് പറയുന്നത് AT റിച്ച് ഡാർക്ക് ബാൻഡ്സിൽ അഡെനിൻ തൈമിൻ will be rich or AT will be rich. Light bands-ലാണെങ്കിൽ GC will be rich. Next is G banding technique. G banding technique is another technique which is used with the help of the stain called GMSA stain. GMSA stain which is stain ചെയ്യുന്നതിനെയാണ് G stain ചെയ്ത് കഴിയുമ്പോൾ കിട്ടുന്ന ബാൻഡിങ് പാറ്റേണിനെയാണ് നമ്മൾ G banding pattern എന്ന് പറയുന്നത്. ഡീറ്റെയിൽസ് നിങ്ങൾക്ക് ഇപ്പം വേണ്ട. Just have a look at what is happening. Then N banding technique. N banding technique, I said NOR region. N banding technique എന്ന് വെച്ചാൽ NOR region structural non-histone proteins linked to NOR region is taken into consideration for the staining. Next is C banding technique. C banding technique is actually done with the GMSA stain itself, but the treatment is different. Chromosome treat ചെയ്ത് കൊണ്ടുവരുന്ന pathway it is slightly different from the Q banding. And these are the 4 basic types of chromosome banding techniques or 4 types of staining technique which is used to study the banding patterns in chromosome. And this C banding is commonly used for insects and plants. C banding is used for insects and plants. Insects and plants. Then N banding. N is used in the case of mostly the most preferable type of staining pattern is the N pattern for the plants. Then, but here G and Q is GMSA staining and Q is used in case of human beings. G and Q type of banding pattern is used for human beings, but the G banding is not used in the case of plants. G is not for, this is used for animals, and the C is not used for plants. Now, next is chromosome band, and as a result of this, the representation of this karyotyping, each chromosome will be having, when we draw, each chromosome will be having a short arm and a long arm. Chromosome will be having a short arm and a long arm, and this short arm is usually represented as P. P means petit. It is small, and the long arm is represented as Q. So chromosome usually there are 2 types of the 2 arms are there. One arm of the chromosome is short, and that is represented as P, denoted as P, and long arm is denoted as Q. For Q means you stand in Q, that, and it is separated by a centromere. And each chromosome is divided into bands, and the bands we get as a result of this staining techniques. We have different bands in the chromosome. This is simply what we get in karyotype. Karyotype ഒരു ക്രോമസോമിന്റെ ഒരാമ നീളം കൂടുതലായിരിക്കും, ഒന്ന് ചെറുതായിരിക്കും. ഇനി നമ്മൾ ഓരോ ക്രോമസോമിന്റെ സ്ട്രക്ചറിലോട്ട് പോകും, അന്നേരം you will be able to understand. So that is the representation of chromosome band. For example, ഇപ്പോൾ ഒരു ചെറിയ എക്സാമ്പിൾ പറയുകയാണെങ്കിൽ, ഞാൻ പറഞ്ഞു ലോങ് ആമും ഉണ്ട് ഷോർട്ട് ആമും ഉണ്ട്. ലോങ് ആമിനെ Q എന്നും ഷോർട്ട് ആമിനെ P എന്നും വിളിക്കുന്നു, ഇല്ല. സോ ദ സൈറ്റോജനറ്റിക് എക്സാമ്പിൾ ഇങ്ങനെ ഒരു നൊട്ടേഷൻ കണ്ടു കഴിഞ്ഞാൽ, ഇഫ് യു സീ എ നൊട്ടേഷൻ ലൈക്ക് ദിസ് 7Q31.2 this means Q means long arm. So this indicates the gene is on the 7th chromosome. The cytogenetic map of the location of gene means The gene is on the chromosome number 7. അതാണ് 7q. 7q എന്ന് പറയുമ്പോൾ gene is located on chromosome number 7. Then ends of the ഇനി q ഏതാമാണ്? 7 എന്ന് പറയുമ്പോൾ ഇപ്പോൾ 7 q 3 1.2 point. ഇപ്പോൾ 7 എന്ന് പറയുമ്പോൾ ആദ്യത്തേതാണ് 7. 7 means 7th chromosome. അല്ലേ? Number 7. ഇനി ഇവിടെ 8 ആയിരുന്നെങ്കിലോ? If it is 8, then that means 8th chromosome. Then Q means long arm. Q arm. Q is the arm of the chromosome. That means the long arm. Then 3 means the band. So 7th chromosome. If this is the 7th chromosome, this shows 7th chromosome. If this is the 7th chromosome, 7th chromosome long arm. Long arm ആണ് Q. അപ്പോൾ long arm 3rd position. അപ്പോൾ ഇതാണ് 1 2 3 4 5 എങ്കിൽ 3rd പൊസിഷനിൽ അതിൻ്റെയും സബ്സെറ്റ് ആയ 1 2 മൂന്നാമത്തെ ലൊക്കേഷനിൽ ഒന്നാമത്തത് വീണ്ടും അതിൻ്റെ സബ്സെറ്റ് ആയ രണ്ടാമത്തെ ലൊക്കേഷൻ. ദാറ്റ് ഇസ് വാട്ട് ഇസ് നോൺ ആസ് cytogenetic map location. This is how you identify. ഇതാണ് ആക്ച്വലി ബാൻഡിങ്ങിന്റെ ബെനിഫിറ്റും അല്ലെങ്കിൽ കാരിയോടൈപ്പിങ്ങിന്റെ ബെനിഫിറ്റ് എന്ന് പറയുന്നത്. വീൽ സെഡ് അല്ലേ, അത് എവിടെയാണ് കാരിയോടൈപ്പിങ്ങിന്റെ അഡ്വാൻറ്റേജ് പെഡിഗ്രി അനാലിസിസിനും എവല്യൂഷണറി സ്റ്റഡിയിലൊക്കെ വരുന്നത് എന്താണ്? നമ്മളൊരു കാര്യം, ഒരു ജീനിന്റെ ലൊക്കേഷനും ഒരു ജീനിന്റെ ഇപ്പൊ ഫോർ എക്സാമ്പിൾ ഇപ്പൊ ജീൻ റെസ്പോൺസിബിൾ ഫോർ ദ കളറേഷൻ ഓഫ് ഐ ഇസ് ലൊക്കേറ്റഡ് അറ്റ് ദ 7th ക്രോമസോമിന് ഇന്ന പൊസിഷനിൽ ഈ മൂന്നാമത്തെ ലൊക്കേഷനിലിരിക്കുന്ന ജീനാണ് കണ്ണിന്റെ കളറിനെ റെഗുലേറ്റ് ചെയ്യുന്ന പറയുന്നത്. അതാണ് കാരിയോടൈപ്പിന്റെ അഡ്വാൻറ്റേജ്. സോ ദാറ്റ് ഷുഡ് ബി കെപ്റ്റ് ഇൻ മൈൻഡ് ഓൾവേസ്. അപ്പോൾ നിങ്ങൾക്ക് ഇപ്പോൾ ക്വാളിറ്റി ഫോർ കോമ്പറ്റേറ്റീവ് എക്സാം ആൻഡ് ഓൾസോ ഇഫ് ദിസ് കംസ് യു ഷുഡ് ബി ഏബിൾ ടു ഇന്റർപ്രറ്റ്. ഇങ്ങനെ ഒരു ക്വസ്റ്റ്യൻ കിട്ടിക്കഴിഞ്ഞാൽ യു ഷുഡ് ബി ഏബിൾ ടു അണ്ടർസ്റ്റാൻഡ്. 7 എന്ന് വെച്ചാൽ ക്രോമസോം നമ്പർ, Q ലോങ് ആം, 3 3rd ബാൻഡ്, ദെൻ അടുത്ത 3 അപ്പൊ ആദ്യം വരുന്നതിന്റെ സബ്സെറ്റുകളായിരിക്കും അതിനുശേഷം വരുന്നത്. ഇനി അടുത്ത ഇമ്പോർട്ടൻ്റ് ആയിട്ട് ഓർത്തിരിക്കേണ്ടത് the ends of the chromosome are labeled PTEL and QTEL. P എന്ന് വെച്ചാൽ ഷോർട്ട് അല്ലേ, ഇതിനകത്ത് P means short and Q means short arm. ഇതിനകത്ത് ഈ Q means long arm. Q means long arm. So the end of the chromosome are always labeled with PTEL. Tel means the tip. അല്ലെങ്കിൽ telomere, I'll tell you, that's the tip of the chromosome. അപ്പോൾ ഷോർട്ട് ആമില് P എന്ന് പറയുന്നത് ഷോർട്ട് ആമിന്റെ ടിപ്പും QTL എന്ന് പറയുന്നത് ലോങ് ആമിന്റെ എൻഡും ആയിരിക്കും. സോ ദ നൊട്ടേഷൻ, ഇഫ് ദ നൊട്ടേഷൻ is 7QTEL അല്ലെങ്കിൽ ഇപ്പൊ 8QTL or 9QTL or 1QTL, anything can be there based on the number of the chromosome. We are having 23 pairs of chromosomes and you can take into consideration either the first chromosome till the 23. ഏത് വേണമെങ്കിലും നമുക്ക് എടുക്കാം. അതിൽ ഇപ്പൊ 7QTL എന്ന് പറയുമ്പോഴേക്കും 7th chromosome, that is QTL refers to the end of the, കാരണം എന്താണ് TL എന്ന്, TL വരുമ്പോൾ end ആണ്, TEL means end. ഇനി Q means long arm, so long arm of chromosome 7. മൂന്ന് കാര്യങ്ങളാണ് ഇവിടെ വരുന്നത്. 7th chromosomeന്റെ long armile end region. That is what is the notation means. Now, next is the International System for Human Cytogenetic Nomenclature. നിങ്ങൾ IUCN അല്ല, this is ISCN. International System for Human Cytogenetic Nomenclature gives the specific loci or the specific number to the chromosome location. Here you can see one subset another. These are the location XP. XP എന്ന് പറഞ്ഞാൽ P, short arm of the chromosome. This is short arm. This is the short arm. And this is the long arm. So it's very clear. ISCN or International System of Human Cytogenetic Nomenclature gives the notations for the representation of each gene on a chromosome. So always the lowest number. ഇവിടുന്ന് സെൻട്രോമിയറിൽ നിന്ന് ഇങ്ങോട്ടാണ് numbering start ചെയ്യുന്നത്. You can see here 1, 2, 3, 1, 2, 3 like that. So this means നമ്മൾ നേരത്തെ എഴുതി ഫസ്റ്റ് ക്രോമസോമിന്റെ സബ്സെറ്റുകളായിരിക്കും 2, 3 ലോട്ടൊക്കെ വരുന്നത്. Then here also 1, 1 and 3. Likewise. Then the highest number will be at the tips and the lowest number will be towards near the centromere. That is what is known as, that is the International System of Human Cytogenetic Nomenclature. Now we will come to the classification of chromosome based on the position of the centromere. So this is a chromosome. Again, I think this I've repeated so many times. I've taken the details of the centromere, chromosome structure, etc. in the mutation section also. The point mutations, structural mutations, aberrations. So this is a chromosome, a pictorial representation of a chromosome. This is a centromere. Centromere holds the two arms of the chromosome together, and these are called the chromatids. Just remember, just recall these terms which we studied. The chromosome, chromatids. The chromatids are held together by the centromere, and the location, genes are located on these chromosomes. Genes or the segment of chromosome is called gene, and inside the gene you can see the chromosomes. Now, next is what a centromere is. I have repeated so many times. The centromere is the constricted region. See, this is the constricted region. What did you do? And centromere. Then centromere is the that is known as centromere, and centromere is the constricted region of the linear chromosome. Then it divides the chromosome into two regions. നമ്മൾ സിംപ്ലി എപ്പോഴും വരയ്ക്കുന്ന പടമാണ് സെൻട്രോമിയർ. ക്രോമസോമിനെ രണ്ടായിട്ട് തിരിക്കുന്നു, രണ്ട് എൻഡ്, two arms ആയിട്ട് divide ചെയ്യുന്നു, and divides the chromosome into two regions, and that is referred to as the arms of the chromosome. And it helps to align the mitotic apparatus during cell division. During cell division, just remember we have told this, during cell division the chromosomes will be aligned at the metaphasic plate, and it will be moving towards the either poles. It will be moving towards either poles, and the spindle fibers will come and attach on this centromere. So it helps to align the mitotic apparatus. So during mitosis, the chromosomes are pulled equally to either sides of the poles when the mitotic spindle attaches on this centromere. Centromere lot vanna mitotic spindles. Evidanna ee mitotic spindle varunathu centrioles il ninnu. Centrioles il ninnu vanna mitotic spindle attach cheyidittu പുള്ളി ചെയ്യുന്നു. പുള്ളിയെന്ന് എങ്ങനെയാണെന്ന് നമ്മൾ പഠിച്ചിട്ടുണ്ട്. Depolymerization വഴി, it constricts and the chromosomes are pulled to either ends of the cell, and it provides a site for the attachment of chromatids. based on the location of the centromere. Centromere സെല്ലിന്റെ ക്രോമസോമിന്റെ എവിടെയാണോ ലൊക്കേറ്റ് ചെയ്തിരിക്കുന്നത് അതിനെ ബേസ് ചെയ്ത് ക്രോമസോം പലതായിട്ട് ഡിവൈഡ് ചെയ്തിട്ടുണ്ട്. This is a very easy portion and you every time you get question for your exams too. It's very easy and simple and based on the location of this chromosome, location of the centromere, the chromosomes are divided into different types. Metacentric chromosome, submetacentric, acrocentric, and telocentric. These are the different types of chromosome based on the location of the centromere. First is metacentric chromosome. Metacentric chromosome is the type of chromosome in which the centromere is at the exact center. Centromere is at the center. You can see the centromere is at the center of the chromosome. Exact center or slightly different. Then submeta. Sub means കുറച്ച് താഴെയോ കുറച്ച് മുകളിലോ അതിനാണ് submetacentric. ഇവിടെ ഇപ്പോൾ കുറച്ച് മുകളിലായിട്ടാണ് കാണിച്ചിരിക്കുന്നത്. See the length difference. Submetacentric. Submetacentric means the centromere is located away from the center towards one end. ഏതെങ്കിലും ഒരു സൈഡിലോട്ട്. അപ്പോൾ എന്ത് പറ്റും? The different sizes, length is different. Submetacentric chromosome ആവുമ്പോഴേക്കും ഒരു P arm you can see and a Q arm you can see clearly. So submetacentric means the location of the centromere will be slightly away from the center towards the end of the chromosome. That's known as submetacentric. Now, next is acrocentric. Acrocentric chromosome means the centromere is located very close to one end. See, it is centromere is located to very close. This difference is length is shortening. This is this much long and this is one arm. So the movement is towards the tip of the chromosome. So in acrocentric chromosome, the centromere is located very close to one end of the chromosome. And next is the telocentric chromosomes. Telocentric chromosome means the centromere will be located at the tip of the chromosome. And this is actually, it's not a stable type of chromosome and this does not exist in a cell. Commonly, telocentric is it's a very unstable form. So it does not exist very common in a cell. So these three are the types of chromosomes based on the location of centromere. Metacentric, submetacentric, acrocentric and telocentric chromosome. This is also another, if the centromere is absent, these are all hypothetical representation, or this is not commonly found in a cell. And this is dicentric if a chromosome is having two. Centromere is dicentric. Acentric means no centromere. Now, metacentric chromosome. In human beings, we have different types of chromosome. That is the centrally placed, submetacentric, acrocentric and telocentric. And I said telocentric, humans don't have this type of chromosome, but it is found in some other species. This telocentric is not existing in a human cell. The next is, you can see a picture. In human karyotype, the chromosome 1 to 3 is having, this is A. 1 to 3 is represented as A. Then 4 and 5 is represented as B. Then 6 to 12 as C. Then 13 to 15 is represented as D. Then 16 to 20 or 16 to 18, it is E. Nineteen to 20 is F. Then 21 and 22 and Y are G. This is how in human karyotype the chromosomes are arranged like this, or some names are given in a karyotype. Why? The purpose is to clearly say if you are speaking about a chromosome number 1, or if you are talking about a chromosome number A in general, these terms can be used. The A set of chromosome, the B set of chromosome, like that. So A, the group A chromosome have large, they are metacentric chromosome and they are large. Like karyotype, I have told the larger one will be at the extreme left and the size decreases. In the decreasing length they are arranged, and the shortest will be at the right end, and the sex chromosome or allosomes will be, these are the allosomes. The allosomes will be placed at the bottom right. These are the allosomes. So 1, 2 and 3 are long metacentric chromosomes. They are the large metacentric chromosomes A. Then 4, 5 are large but they are submetacentric. Here you can see they are submetacentric. These are metacentric chromosomes. B, the 4 and 5 are submetacentric chromosomes. Then 6 to 12, they are medium size, they are small, they are not large. They compared to this chromosome number 1, they are medium sized, but they are submetacentric chromosomes. Then coming to the chromosome number C, they are medium sized and submetacentric chromosome. You can see a long Q and a short P arm here. So they are submetacentric medium sized chromosome from 6 to 12. And the chromosome, sex chromosome X come along with this. That is almost the length and the pattern is almost same as that of the chromosome set C. Now, chromosome D are medium sized chromosome. These are medium sized chromosome. They are medium sized chromosome, and these chromosomes are also medium sized. Medium sized. These are long. A will be very long, or the longest chromosome. A are the long chromosome. Then D are medium sized and acrocentric. Medium sized and acrocentric chromosome. Medium sized and acrocentric chromosomes. Then E and F. E are small. E are small chromosomes. Then F is also small. This is metacentric and submetacentric. These are metacentric and this is submetacentric chromosome. Then G, it is small acrocentric. G is also small chromosome and it is acrocentric. The centromere is towards the tip of the chromosome. These are acrocentric chromosomes. So this is a general account of karyotype, or the arrangement of 23 pairs of chromosomes in human beings. So long A, Then a little bit large, metacentric are B, C, medium sized with submetacentric, then D, medium acrocentric chromosome, E, small chromosomes, it can be metacentric or submetacentric, then F are small metacentric chromosomes. Then G are small acrocentric chromosomes and C and G are the allosomes. Now, next is the pedigree analysis. In this pedigree analysis also we use for pedigree analysis we use this karyotyping. Karyotype, radiogram, etc. are used in pedigree analysis. What pedigree analysis is? Pedigree analysis is the application of Mendelian principles. The basic in genetics is the application of Mendelian principles. Mendelian principles are applied basically for the pedigree analysis. In pedigree analysis is used, it's used for studying human inherited diseases. Then it is easy for finding the relationship between the families, and they are always used to determine the mode of inheritance, that is whether a character is dominant, whether a character is recessive, and the genetic diseases, gene associated syndromes, etc. So pedigree analysis is very important. It is used for the study of human inherited diseases. In case of human beings, it's not only applied to human beings, in any organisms, in plants, in cattle, in any organism, you can predict the inheritance pattern in all the organisms. That is pedigree analysis. And these are some of the highlights of why it's being used. So it's used to determine the mode of inheritance. We can say whether a character is dominant, whether a character is recessive, etc. And this is actually widely used in genetic counseling. The main application of this pedigree analysis is its use in genetic counseling. It is being used in genetic counseling. We know again it is widely used in genetic counseling. So the why when this pedigree analysis is being used, because experimental matings, we cannot say a two-person in what animal, in human beings, experimental matings cannot be done. So pictorially we can represent and find how far the diseases can be inherited. Then duration required for multiple generations are very easy. So if we have a family history of some specific disease, we have a family history of one specific disease, we can draw the inheritance pattern from the forefather, from the grandparents till the grandchildren, and find how much is the chance of that disease to be inherited to the offsprings. That is one application. And they show the symptoms of genetic disorders. If a person or if a child in a family is showing some genetic disorder, the history can be tracked, and they are concerned about passing a genetic problem to a children, whether it is passed on from parent to offspring. So it is widely used, or the pedigree analysis has a main application in genetic counseling, because the genetic counselors, when two people who wish to get married or to have children approach a genetic counselor, he can ask about both the positive traits, the genetic disorders or anything, and can to some extent predict how far or this some of the characters can be inherited, both positive and negative. So this is widely used in pedigree analysis. And it is a construction of a family tree. Actually, pedigree analysis is simply a construction. It looks like a tree, like this. It goes like this. It looks like a tree, or looks like a hierarchy. Then it is. So, pedigree analysis is the construction of a family tree based on the careful compilation of phenotypic records. That is very important. Always the counselor should keep in mind the phenotypic records of the family. That is genetic counseling. That is pedigree analysis. So, pedigree analysis is the construction of a family tree based on the careful compilation of phenotypic traits or records of the family over several generations. ഇല്ലേ ഒരാളോട് ചോദിക്കാണ് now the thing which is very common is ഇല്ലേ it's not being maybe may not be used by doctors. What people now commonly use is if a person is telling yeah he is having diabetes or he is having some other problem, the common question which even the layman asks is, oh, in family anybody is having this disorder, that means this certain diseases are inherited from the parents. പലരും വീട്ടിലൊക്കെ you might have heard people asking വീട്ടിൽ ആർക്കെങ്കിലും ഉണ്ടോ എന്ന് ചോദിക്കുന്നത് എന്താണ് ഫാമിലി ഹിസ്റ്ററിയിൽ ഉണ്ടോ എന്നറിയുന്നത്. അതിന് വേണ്ടിയിട്ടാണ് നമ്മൾ പെഡിഗ്രി അനാലിസിസ് എന്ന് സിംപ്ലി പറയുന്നത്. സിംപ്ലി ഇപ്പോൾ അച്ഛൻ്റെയും അമ്മയുടെയും ബോത്ത് ഫാദർ ആൻഡ് പേരൻസ് സൈഡിൽ മുതൽ മുകളിലോട്ടുള്ള ട്രീ നമുക്ക് ട്രേസ് ചെയ്യാം. എങ്ങനെയൊക്കെ ഇത് വരാം. And this are some of the ഇങ്ങനെ ആ ട്രീ വരയ്ക്കാനായിട്ട് we use some shapes. We use some shapes to draw this pedigree chart. I said pedigree analysis എന്ന് പറഞ്ഞാൽ simply a construction of a family tree. അപ്പോൾ ആ ഫാമിലി ട്രീ ചുമ്മാ വരയ്ക്കാൻ പറ്റത്തില്ല. അതിനകത്ത് it's a universal symbol. There are some universal shapes which is used to make a pedigree chart. കാരണം pedigree chart ഒരാൾ വരച്ചത് വേറൊരാൾ കണ്ടു കഴിഞ്ഞാൽ അയാൾക്ക് മനസ്സിലാകണം. That is very important. Genetic counselors ആണെങ്കിലും pedigree analysisന് വരച്ചു കഴിഞ്ഞാൽ ഇപ്പോൾ these are the universal shapes. These are the universal shapes which is used in pedigree analysis. If it is square stands for male, അല്ലേ father ആണെങ്കിൽ male. ഇനി affected male എന്തെങ്കിലും ഒരു അസുഖം, simply if you say diabetes, simply affected male. Affected male is ആണെങ്കിൽ ബ്ലാക്ക് കളറിൽ റെപ്രസെന്റ് ചെയ്യും. ഇപ്പോൾ ഒരു ഏതെങ്കിലും ഒരു ഡിസീസിന് ബേസ് ചെയ്താണ് നമ്മൾ പെഡിഗ്രി അനാലിസിസ് ചെയ്യുന്നതെങ്കിൽ if you are using this pedigree analysis for a specific disorder, affected male നെ ഇങ്ങനെ കാണിക്കും. For example, if you are doing baldness എന്ന് വെക്കുക. അപ്പോൾ ബാൾനെസ് ഉള്ള മെയിലിനെ എന്താണ് ബ്ലാക്ക് ആയിട്ട് കാണിക്കും. ഫിനോട്ടിപ്പിക്കലി എക്സ്പ്രസ് ചെയ്യുന്നത്. ദാറ്റ് ഇസ് വാട്ട് ഇസ് ബ്ലാക്ക് കളർ. ഇനി ഫീമെയിലിനെ ആണെങ്കിൽ സർക്കിൾ ഷേപ്പ് ആണ് യൂസ് ചെയ്യുന്നത്. അഫക്റ്റഡ് ഫീമെയിൽ ആണെങ്കിൽ ഇറ്റ് വിൽ ബി കളേർഡ്. മീൻസ് നോട്ട് കളേർഡ്, എന്താണ് ബ്ലാക്ക് കളറിൽ ദാറ്റ് ബോക്സ് ഓർ ദ സർക്കിൾ വിൽ ബി filled. That is affected female. If it is unknown sex is not determined properly, the symbol which is used is a rhomboid shape. And if there is a chance for the offspring to die, and the symbol which is used is this. These are the common symbols which is used in the drawing the pedigree chart. Now, and if a single line is shown between the male and the female, this is male and this is female. If a single line is drawn between the male and female, that represents mating. This line represents mating. Now, if it is represented by a double line means consanguineous mating. Consanguineous means the relationship between the relatives, that is It shows consanguineous means if two persons are having the same genetic history. That means relatives. Simply നിങ്ങൾക്ക് മനസ്സിലാകാൻ വേണ്ടിയിട്ട് ഇപ്പം നമ്മുടെ കേരളയിലുള്ള ഒരു സിറ്റുവേഷൻ എന്താണ് മുറച്ചെറുക്കൻ എന്ന് പറയുന്നത്. That is consanguineous mating, or that is between the if there is a inheritance or both the parents are having common ancestors, that is known as consanguineous mating. And if for offsprings, a line like this, and the arrow points are used to highlight which organism or which individual, and this is commonly this is what is known as pedigree chart. Also, pedigree chart shows the inheritance pattern of a particular trait that is governed by a gene or a group of genes, and the gene symbols are used in the pedigree to predict, and this, by drawing this, we can predict the inheritance pattern. And simply by drawing this, you can say if affected male, or if affected male, we have learned this in chromosomal aberrations and all. We have learned about baldness, different genetic disorders and all. If you're drawing like this, and they're having, if a father who is having baldness is marrying a normal lady, and they are having one boy and two boys, one boy is having baldness, then this is simple. This is a simple one. Actually, this is not used, bald, and here this shows one of the offsprings who are males are of this. If these two parents are having two sons, there is a chances like this. This is how we simply show the inheritance pattern with the help of a pedigree chart. Now, applications of pedigree analysis. Where is this commonly applied? Pedigree analysis is used in, I said, 1. Genetic counseling. The genetic counselors use to predict the probability of inheritance pattern into the children. Then it is used in undertaking breeding experiments in animals. These two are the most commonly applied areas of pedigree analysis. Pedigree analysis is used in genetic counseling, and 2, it is used in undertaking breeding experiments in animals. Now, next is genetic disorders in man. Before going into the genetic disorders of man, let me tell a few details. That is allele. We have discussed this many times, and you can even recall what I have taught in this mutation section also. So, allele is an alternative form of a gene. Just few terms. Alternate form of a gene is called allele. Then, alleles can be homozygous or heterozygous. Homozygous means two same alleles, which is meant for a character. Heterozygous means two different alleles. Then, usually in inheritance, a dominant is crossed with a recessive one, the resultant will be dominant. A dominant with a dominant, the trait will be dominant. Then recessive plus dominant gives a dominance. Recessive plus recessive gives a recessive condition. So the expression of recessive is 3:1. This is how we have learned in Mendelian genetics, that is 3:1. Only if recessive character come in two genes or in homozygous condition, that will be expressed. That is homozygous, just keep in mind what homozygous is, what heterozygous is, what recessive is, what dominant is, and When mixing or when a dominant is crossed with a recessive one, result in a dominant. Dominant cross with a dominant result in dominant, and this is one example. Capital B means heterozygous. Sorry. Capital B means homozygous. I'll go to the next slide. Yeah. Capital B shows a homozygous. Capital B, small b shows homozygous. This is capital B is homozygous condition. Then capital B, small b is heterozygous. Then small b is recessive. These are some of the terms. Now we'll go to the categories of inheritance. The categories of inheritance, it can be, that is, there are different types of inheritance pattern or genetic disorders in man, may be due to chromosomal anomalies, that is, if there is a change in the number of chromosomes, or if there is a single gene anomaly or single gene disorders are there in the genetic disorders of man. So these are, if there is a change in the number of chromosome of the autosome, it means autosomal anomaly. Autosomal anomaly can be autosomal recessive type or autosomal dominant types are there. If it is sex-linked, it is X-linked, recessive, X-linked, dominant, then Y-linked. These are some of the common disorders which is or genetic disorders associated with human beings. And now we'll look into it one by one. First one is the let me discuss about, we'll talk about one and we'll conclude. And this is a inheritance pattern, how we draw. If this is a heterozygous, that is a homozygous, and the inheritance pattern is 50%. This is heterozygous and homozygous related with the eye color. Now, the chromosomes are of different types. Chromosomes, there are sex chromosomes and autosomes. Chromosomes are of two types. There can be sex chromosome and there can be autosomes. In chromosomal mutations, we said chromosome mutation can be due to deletion, due to inversion, can be due to duplication, due to inversion, or due to non-disjunction. ഇതിലെ ഏതെങ്കിലും കാരണം വഴി എന്താണ് mutations can result. So first is disorders due chromosomal anomalies, that is change in the chromosome number. Chromosome number. Gene mutation can be either inherited or acquired, and a hereditary mutation is a mistake that represents in the DNA of virtually all body cells, and that is known as germline mutation. And the genetic disorders of man probably will continue in the next class. Hope you understood what is being taught in today's class. So in today's class we discussed about the chromosome, types of chromosome, the banding pattern, pedigree analysis and applications of pedigree analysis. And thank you for your listening.